Good evening, Snehanshu. Good evening, Ovirup. Okay, hold on for a second, okay? Okay, hold on for a second, okay? Good evening, Suraksha. Everything is clear. Picture, sound. Okay. Thank you for your feedback. I'll just, begin it. So, we'll move forward with photosynthesis. We'll move forward with photosynthesis. Yeah, so this is it. Now, okay, I think, yeah, fine. So, fine. So, yeah, talking about photosynthesis, when we try to talk about photosynthesis, things that are there are pigments, things that are there are light. Okay, so what is photosynthesis? Photosynthesis is making of glucose, or we can say starch, with the help of light, water. Light means sunlight, water, and also chlorophyll. So these are certain things that are required in photosynthesis, Now, we have in the last class, in the last video, we have talked about, in the last video, we sure have talked about what? We have talked about, as I know, is about the structure of chlorophyll. That is a photosynthetic pigment. And also, I taught you the difference between chlorophyll A and chlorophyll B. Also, I taught you the difference between chlorophyll A and chlorophyll B. So, this is what I have taught you. You remember the R group, one has the CH3, the other has the CHO. So, and hence they were different. Also, there were 7 types of chlorophyll. Chlorophyll A, B, C, D, E. I think 7 types of chlorophylls were there. and they were wait, hold on. That's great. Just hold on for a second. Yeah, ABC, sorry, 5 types of chlorophyll were there. There were 5 types of chlorophyll. Okay, so and the other pigments that you should know about. Now let's begin the class in a full-fledged thing. Good evening, Nikita. The other pigments that we should talk about are the 5 carotenoids. Carotenoids. This is one pigment, carotenoids. Okay, and also we should talk about xanthophyll. So these, I think I have talked about these pigments beforehand also, okay, while teaching cells. But let us today in detail talk about, not in detail, these carots. Look, in the process of photosynthesis, chlorophyll is the main pigment. Okay, in fact chlorophyll A is the main pigment, which we will learn shortly, that why I am saying, or which discovery led us to know that chlorophyll A is the main pigment, that we will learn shortly. But the fact is this, that yeah, okay, chlorophyll A, then B, C, D, E, this carotenoids, xanthophylls, okay, and in some algae, especially the algae means what? The photosynthetic blue-green algae. Algae are all photosynthetic. In blue-green algae, we do find another type of what we can say pigment, that is known as phycobilins. Okay, that is known as phycobilins. So that also you should keep in your mind. So these are the things. Now, if I talk about carotenoids, and I talk about xanthophyll. If I talk about carotenoids and xanthophylls, we see that the formula of carotenoid or carotene is C40H56. C40H56. However, the formula of xanthophyll is C40H56O2. Okay, the color of carotenoid is orange-red. Okay, however, the color of xanthophyll is yellowish-brown. The color of xanthophyll is yellowish-brown. So this is the thing that you should keep in your mind, that or keep it, and the most common carotenoids that we find, or sorry, most common xanthophylls, the most common xanthophylls that we find are fucoxanthin or lutein. Fucoxanthin or lutein. Look, these notes should be written by you, okay? It's very important that you write down the notes. Lot of people, I don't know, only few of you send me notes. But in class 11 and 12, writing notes is very important, okay? Otherwise you will not remember these things. So that is the thing. So we have this. Now, moving forward, I have been today started to talk about in the last video I have talked about the chlorophyll. There are various types of chlorophylls. I have talked about chlorophyll A and B and the difference and what is the difference. Various types of difference were there. All these things I have talked about. And there are also some other pigments called carotenoids and xanthophylls, and I have talked about carotenoids and xanthophylls. Also, in blue-green algae, you find phycobilins. Now, moving forward with our discussion, see, what happens is that if I talk about phycobilin little bit, phycobilins, if I talk about phycobilins a little bit, now the fact is this, that phycobilins are found in blue-green algae, that you know. There are two types of phycobilins, the phycocyanin the phycocyanin, which is purple in color, and the phycoerythrin, which is red in color. Okay, now remember, in phycobilins there is no magnesium in the middle or no phytol chain. There is no magnesium or no phytol chain. What are the most common carotenoids you find out, Sneha? So this I don't know. Okay, I have not found this. You know, I have seen. I'm teaching you according to the questions. So this I have researched this only. Okay, you find out and tell me which are the most common carotenoids. Carotenoids are basically found in fruit peels. Okay, so they are also photosynthetic pigments. Xanthophyll are also photosynthetic pigment. That is a different thing. Okay. Now the fact is that when you move forward, phycobilins. No, in phycobilins there is this phycocyanin and phycoxanthin. Two types are there. Phycoerythrin, and no magnesium and no phytol chain is present in phycobilins. However, in carotenoids also you will find phytol chains, and Okay, and in Xanthophyll also you will find phytol chains or magnesium ions, but magnesium in the center. But in phycobilins you will not find it. So that you should keep in your mind. Now moving forward, you see, remember, this I am talking about the carotenoids, I am talking about the xanthophylls, I am talking about the phycobilins, I am talking about chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E. See, except chlorophyll A, okay, except chlorophyll A, which acts as the reaction center, I must say. Now you will question me, what is the reaction center? You can imagine that reaction center is a place where the process of photosynthesis or the process of light capture or light reaction of photosynthesis begins. That is the reaction center. All the energy from light captured by all other pigments, okay, all the energy from light captured by all other pigments is transferred to reaction center. Okay, suppose for example there are, like this kind of thing is there. Suppose lots of accessory pigments are there. These are accessory pigments. In the middle there is chlorophyll A. This chlorophyll A becomes the reaction center. And these all other pigments I'm talking about, all other pigments which transfers the energy to the chlorophyll A or the reaction center are known as accessory pigments. Okay, so this is the thing that I wanted to say you. So I hope that you have understood this particular thing that I have said. Just hold on for a second. Tell me, is it visible now? Is it visible? Please tell me. Okay, thank you, Abhidu. Hold on for a second. Okay. Sorry, I was having tea. I'm so sorry. Anyway, so like we see that now. Okay, let me tell in much more detail now. So we have understood this, and we have understood this also, that okay, these are called accessory pigments. Who are called accessory pigments? All of the pigments. All chlorophyll B is accessory pigment. Oh, sorry, chlorophyll. Chlorophyll B is accessory pigment. Chlorophyll C is accessory pigment. Chlorophyll D is accessory pigment. Chlorophyll E is accessory pigment. Carotenoid is accessory pigment. Xanthophyll is accessory pigment. Phycobilin I will not tell accessory pigment. Remember this. Many students does the wrong. Phycobilin cannot be accessory pigment. Phycobilin is a pigment which is present in blue-green algae. However, in blue-green algae, chlorophylls are also present, but it is an accessory pigment of blue-green algae. It is not accessory pigment of higher plants. Not accessory pigment of higher plant. Remember that. Not the accessory pigment of higher plant. It is the accessory pigment of blue-green algae. But however, if they ask you in question, what is the accessory pigment of higher plants? B, C, D, E, carotenoid, xanthophyll. These are the accessory pigments of the higher plants. So that you should keep in your mind. Okay, so these are the things. Okay, so we have learned about the the chlorophyll that is there, and we have learned about that chlorophyll A is the main pigment or main photosynthetic pigment which forms the reaction center. Other all pigments do form the accessory pigments. They are the accessory pigments, and they transfer their light energy that they have captured to the reaction center or to the chlorophyll A. Fine, you know this from me. Now moving forward with more with our discussion, what are we ought to say or what can we say? Okay, just let me hold on for a second again. Let me see this thing. Okay, hello. Okay, now tell though, is the thing visible? Is the thing fine or not? Wait, hold on. Better. This is the best way, I guess. Okay. Okay, I hope this is the best way we can teach. So anyway, so we have learned about lot of pigments. Now moving forward, now let us. I was really perplexed with my setup. Fine. Now actually, what I want to keep my both hands free. Okay, because for up till now, what? Not visible? Please, everyone, say everything is visible or not. Can you see my writings? Can you see things? I know this one is better. Ovirup, what's the spelling of visible? Are you illiterate? Okay, so we have learnt about all these things. Now moving forward, you know, now moving forward, what happens is that, you see, we will learn about the site of photosynthesis. Now let's do this. This is interesting. The site of photosynthesis. Previously, what used to happen, you know, previously I used to hold the phone in my hand. It used to shake a lot. So better than a shaky video, what we can say, I thought that my hand can also rest and something like that. Okay. So now moving forward, the site of photosynthesis that we are talking about. Okay. So we are talking about the site of photosynthesis. You all know the site of photosynthesis. The photosynthesis has two reactions. Photosynthesis, in class 10, you must have learned that photosynthesis comprises of two types of reactions. One is known as the light dependent reaction, and the other is known as light independent reaction. So one is light dependent reaction, one is light independent reaction. Now when we talk about light dependent and light independent reaction, surely, okay, light dependent reaction, you have learned this in class 10 only, that light dependent reaction occurs in the grana. Okay, but light independent reaction or dark reaction, as we called it, occurs in the stroma. So this is, we know this already we have understood, we know all these things, right? So already we have understood and we know all these things. But the fact is this, you see, still a little bit of recapitulation is required because it is there in your syllabus. Little bit of recapitulation we can do. Now suppose, for example, suppose this is a leaf. Okay, now if you do a transverse cross section of the leaf, the layers of the cell in the leaf that you will find is like this. At the top there will be epidermis and there will be a bottom epidermis. However, there will be presence of stomata in the bottom layer, okay, which you know. There will be presence of stomata in the bottom layer. Now, there will be cuticle above this layer, above the epidermal layer. There will be cuticle in the top epidermis, in the upper epidermis. In the lower epidermis, there will be what we can say stomata or pores. You know about stomata, that there are guard cells. Now what happens in between these two layers, this epidermal layers, suppose this is the epidermal layer, okay, in between these two epidermal layers, what happens is that there are cells. Now the cells that are there are like this, tightly packed near the epidermis, and these tightly packed near the epidermis, and these are called the palisade layer of mesophyll cells. Palisade layer of mesophyll cells. Okay, so mesophyll cells are those cells. Now question arises, what are mesophyll cells? Mesophyll cells are those cells which contain the chloroplasts. Now you know that chloroplasts are photosynthetic organelles. Photosynthetic organelles. Can you see? I hope you can. Okay, are photosynthetic organelles. That we know. Now, these mesophyll cells are the carriers. Suppose if I draw one mesophyll cells, there will be lots and lots of chloroplast inside a mesophyll cell. Remember that. Lots of chloroplasts are there. About 50 to 70 at an average chloroplasts are present in one mesophyll cell. About 50 to 70 chloroplasts are present in one mesophyll cell. Okay, so that you should keep in your mind. Okay, now this mesophyll cells forms a palisade layer, which is a very compact layer. Now below the palisade layer there is another layer which is called the spongy layer. It is like this. Okay, now it is little bit quite unorganized and not very tightly packed. So this, the bottom layer, is known as spongy layer of mesophyll cells. So there is palisade layer of mesophyll cells, and then there is spongy layer of mesophyll cells inside the transverse section of the leaf. Now that is it. Now in the spongy mesophyll layers there will be space. There will be space in between, air gaps, or space for if you cut it. If there is, these are the vascular bundles. These are what? The vascular bundles. If you cut open, you will find vascular bundles through the veins, right or wrong. Here if the thing is there, lots and lots of veins are there, and vascular bundles run through the veins. When you know that vascular bundles run through the veins, okay, so through the veins vascular bundles will run. Okay, and what will happen is that, okay, fine, that vascular bundles are running through the veins, and you will find the vascular bundles also inside in the spongy layer. Okay, not in the palisade layer. That you should keep in your mind. Now, if you have understood the transverse section of the leaf, so what do you understand? That in the mesophyll cells, in the leaf, there are palisade layer of mesophyll cells and the spongy layer of mesophyll cells, which carry 50 to 70 chloroplasts, and these chloroplasts are there inside the, what we can say, the mesophyll cells. And you very well know that grana and stroma are the part of chlorophyll. Okay, that you have learnt in class 10. You see, suppose for example, if you draw a chloroplast, suppose suppose a cell. Now if the cell is there, suppose there is a chloroplast, okay, or there is another chloroplast, and if you take out the chloroplast and magnify this, let me magnify this chloroplast. Suppose this is the chloroplast. Now you very well know that inside the chloroplast there are thylakoid membranes which form stack one over the other. These are the thylakoid membranes which forms stack one over the other, right? When it forms stack one over the other, and these stacks are connected in this way through stroma lamellae. So these, one is known as thylakoid membrane. Okay, and the thylakoid membranes are connected by stroma lamellae. These are known as stroma lamellae. The thylakoid membranes are connected, and they are connected by stroma lamellae. Now the thylakoid membrane is embedded in a jelly-like substance or jelly-like environment entirely, which is the stroma, which is known as the stroma. Now this is the thing that we already know. Hold on for a second. Okay, so this is the thing that we already know. Okay, now if we go much more deeper, if we pick one thylakoid membrane and if we bring it down, you know that inside the thylakoid membrane there are chlorophyll pigment is present inside the thylakoid membrane. The chlorophyll pigments, the structure that you have learned, are present inside the thylakoid membranes. Okay, so the chlorophyll structure is the chlorophyll pigments or the other photosynthetic pigments also are present in the thylakoid membrane. Not only chlorophyll, it's chlorophyll A, chlorophyll B, chlorophyll C, D, E, carotenoid, xanthophyll, all are present in the or inside the thylakoid, and hence they form an entire stack. The entire stack is known as the grana, and each of the grana is connected by stroma lamellae. So this is the details of the structure. Now, as because light dependent reaction is a reaction which is, see, what is light dependent reaction? Light dependent reaction means capturing the light energy. In simple language, if I say capturing the light energy or harnessing the light energy or converting the light energy into reduction potential. If you want to use a little bit, a little bit more scientific language, then you should say that conversion of the light energy into reduction potential is known as the light dependent reaction. Why I am saying reduction potential? Because then you can use this reduction potential, or you can use this energy later to convert it into chemical energy. Okay, reduction potential is itself a chemical energy. You know from chemistry that electricity is the flow of electrons, and electricity and chemical reaction is electricity and chemical reaction is connected to each other by what? Electricity and chemical reaction is connected to each other by this oxidation and reduction reaction. Right or wrong? Movement of electrons is that. So I hope that you understand that capturing of light energy is actually excitation of electrons, which will go into details. I will go into the details of it. So you don't need to worry about it here. But yeah, I am saying that capturing of light energy, converting into reduction potential, that is the light dependent reaction. So definitely, if it is capturing of light energy, definitely it is going to happen in the grana. But the light independent reaction occurs in the stroma, which does not require any light. Light independent means does not require any light. Okay, here what will happen? Here the light independent reaction, that means the reduction potential that has been generated from the light reaction, okay, will be used to produce glucose molecule or starch. Many glucose binded together is known as starch. This is the polymeric form, this is the monomeric form that you know. Okay, so glucose molecule of the starch. So the reduction potential will be used. So that is light independent reaction. Those were my class 10 students. I told them the difference of the timing between light dependent reaction and light independent reaction is 0.001 second. Immediately it occurs. It does captures, creates the reduction potential, creates the, produces glucose or starch. So immediately it kind of happens. Are you getting my point? So kind of it immediately happens. But one requires light, one doesn't require light. So definitely the light independent reaction occurs in the stroma. And this is the place where it occurs. So that is all about the site of photosynthesis. So I hope that you have understood. Now moving forward. With our discussion, let's see. Let us talk about another factor. So what have we talked about? We have talked about, I think, up till now, discoveries. Number 1, done. Number 2, we have talked about pigments. Number 3, we have talked about site of photosynthesis. That is structure of chloroplast. The structure of chloroplast is also you have learned in class cell chapter also. So site of photosynthesis we have learned about. So number 4, now we will talk about another factor, which is light. When we talk about light, we will basically here, later on we will talk about the intensity or amount of light. Here basically we will talk about the quality of light and not quantity of light. Okay, quantity of light later on when we will understand factors which affects photosynthesis. Under that topic we will understand the quantity of light or the intensity of the light or the amount of the light, as you can say. But here we will try to understand about the quality of the light. And when I say the quality of the light, that means the wavelength of the light. Because you definitely very well know that the visible light, if broken down into spectrum, breaks down into 7 colors, which are violet, indigo, blue, green, yellow, orange and red, Vibgyor, which is increasing in wavelength. Means it has the lowest wavelength, which is of 400 nanometer, and this is of around 700 nanometer, which you know, right? Now the fact is this, we will try to talk about that in this range, in which light photosynthesis occurs the most, or which, if anybody asks me the question, that which of the wavelength, lambda, or color of the light, because different colors have different wavelengths, affects the photosynthesis, affects or enhances or light affects the photosynthesis the most. Affect is not English teacher will scold me if I use the affect word here. I should use, what should I use? So the which of the lambda, color of the light, is most efficient in photosynthesis. Yes, is most efficient in capturing light. In doing photosynthesis. This can be the thing. मैंने कौन लाइट है और कौन सा लाइट में photosynthesis ज़्यादा होता है. So that is the basic thing that we will understand about or learn about in this. Now moving forward with our discussion. Okay, so this is what is all about. Now moving forward with our discussion. Now I want to throw you a question. I want to say you that if you were a scientist and you didn't know the answer of it, that out of this VIBGYOR, which light does or affects the photosynthesis the most, or helps in photosynthesis the most, or is useful for photosynthesis the most, you find out what technique or what experiment would you have designed to solve this. Those who are seeing the video, can you think and tell me, or those who is later seeing the video afterwards, can you pause the video? Of course I will tell you the answer. Can you later on tell me that is there any method or way by which I can determine this? Any put pressure in your brain? Is there any method or way, anything that I could do to find out this? Any experimental setup that you can think of? Any experimental setup that you can think of? Would you like to comment and say? First splitting the light, casting them on the, yes, Snehanshu, splitting the light, then casting them on the plant, and then finding out the, what, how will you know the rate of photosynthesis is high, Snehanshu? Okay, fine, you split the light, casting them on the same plant, one by one, day 1, day 2, day 3 karke. But how will you know that the rate of photosynthesis is high? That thing, how will you come to know? That thing, how will you come to know? Yeah, of course, splitting the white light, Snehanshu, but how will you come to know that in which light the photosynthesis is occurring the most? Okay, if you have already thought, let me tell you what is to be done. Okay, let me tell you what is to be done little bit. First of all, There was an experiment I already told. We can check the amount of glucose produced, we can check the amount of glucose produced, and I cannot read the comments because of this holder. Anyway, we can check the amount of glucose produced. Fine, I can understand what you wrote. You see, first, most easiest way, first of all, I would like to say, easiest way to know the rate of photosynthesis. Rate of photosynthesis means not how large amount of photosynthesis is occurring. তাহলে তো ग्लूकोज কত प्रोड्यूस হচ্ছে অনেকক্ষণ ধরে फोटोसिंथेसिस बहुत टाइम लेके फोटोसिंथेसिस कर रहा है या बहुत कम टाइम लेके फोटोसिंथेसिस कर, वो नहीं हो रहा है. रेट ऑफ फोटोसिंथेसिस मींस आई एम टॉकिंग अबाउट स्पीड ऑफ फोटोसिंथेसिस. रिमेंबर दैट, नॉट द अमाउंट ऑफ फोटोसिंथेसिस. थोड़ा हो ज्यादा हो, कितना स्पीड में हो रहा है, first of all. And the speed of photosynthesis can be determined by the determination of rate of release of O2 or release of amount of O2. This you keep in mind. যত বেশি oxygen is a byproduct of photosynthesis, right? Oxygen is a byproduct of photosynthesis. Now, as because oxygen is a byproduct of photosynthesis, so what happens is that the more the rate of release of oxygen, more is the rate of the photosynthesis. First, or the amount of oxygen also you see more, then you can see what to say, the rate of photosynthesis is more. Now, at first, an experiment was done by Engelmann. This I told you earlier also. Again, I should tell here, so that's why I'm telling. Engelmann did an experiment. What he did is, he took an alga known as Cladospora, and he applied VIBGYOR, splitting the light. He applied the light in the tube. He saw that the cladospora was already there. Okay, obviously he saw that in the blue region there was more bubbling. So bubbling means what? Oxygen is releasing, oxygen gas is releasing, that's why it is bubbling. And there was more bubbling in the red region. So the blue region and the red region is showing more bubbles. So that definitely means, or that sure does mean, that rate of photosynthesis is higher. Yeah, so that sure does means that the rate of photosynthesis is higher. Where is the rate of photosynthesis? You see, in the other regions also little bit bubbles were found, but they were nearly negligible to the amount of bubbles which were found here. So we can conclude here that okay, in the blue region and the red region the rate of photosynthesis is high. Now keep this information in one corner of your brain. Now listen to this. I will also make a graph, okay, and I will expose all the colors from 400 nanometer to 700 nanometer. Let it be 500 nanometer, let it be 600 nanometer, and in between all the wavelengths are there. 401, 402, 403, 404, 502, 510, all are there in between. And that I will try to plot the behavior of different chlorophyll pigments. I will try to plot the behavior of different chlorophyll pigments. So this experiment told us that okay, this and this are the best photosynthetic lights which are best in doing photosynthesis. These are most absorbed ones. So this one became clear, and this created to another, this experiment gave rise to another graph. Let me tell you this graph at first. So if I plot VIB-GYOR, actually here also I have plotted VIB-GYOR only. Okay, so if I would have plot, then what is happening, that the rate of photosynthesis is little bit increasing at the blue part, and then it is less and increasing at the red part. Okay, so at the blue part and at the red part, there is more release of O2, so rate of photosynthesis is high. Yeah, that is one thing, that is the sure thing, fine. But the fact is this, that okay, the rate of photosynthesis is high, we agree to the fact. Now we will do, now with this we will learn another thing, that okay, my answer, I hope the question that I had asked and Snehanshu had answered, okay, I think the answer is also clear now. But we will also do another thing, that we will check the behavior of, suppose, chlorophyll A. So we will check the behavior of chlorophyll A. So when we check the behavior of chlorophyll A, we see that the maximum, this graph is known as absorption. This graph is derived from absorption spectrophotometry. Now if you remember, in biomolecule chapter I told you what is spectrophotometry. I don't know how many of you remember that spectrophotometry can determine the light absorbed and light emitted. Okay, so obviously if I plot in the terms of absorbed for chlorophyll A, the region of absorbance was like this. Okay, the region of absorbance was like this. Now when we tell about chlorophyll B, in chlorophyll B the region of absorbance was slightly like this, means slightly little more at a wavelength little more than that of chlorophyll A. You see, chlorophyll A absorbed the blue light the best, you can see here, and the chlorophyll B done in red absorbed a little more wavelength than blue, between blue and green. Chlorophyll A also absorbed the red light the best, and chlorophyll B absorbed a little more than that. Now if I talk about carotenoids, carotenoids absorption spectrum was like this. Okay, so this was the absorption spectrum shown by carotenoid, means most absorption was at the yellow region. Okay, and if I talk about the phycobilins, then in case of phycobilins also we found something like carotenoid only. Okay, so this was the graph. Now if you compare this graph, which is the absorption spectrophotometric graph, and this graph is known as action spectrum graph. Why is it called action spectrum graph? This is called action spectrum graph because here we originally see that the blue and the red is affecting or having an action most effective on the rate of photosynthesis, and this is absorption spectrophotometric graph because here we have just investigated that which of the, what we can say, chlorophyll pigment, or which of the photosynthetic pigment absorbs what color the most. I am not concerned about the rate of photosynthesis here. Here I have just extracted by centrifugation methods or chromatographic methods. What did I say? Centrifugation methods and chromatographic methods, means separation of compounds. As if you remember in class 9, separation of mixture, you have learned centrifugation, chromatography. Okay, so by centrifugation, mainly by chromatography. Okay, so chlorophyll A, chlorophyll B, carotenoids, phycobilins were all separated, and they were put into test tubes, and they were put into spectrophotometer, and all the range of wavelengths were made to fall on them, and it was found that which one was absorbed the most. It was calculated. So if these graphs tell us that chlorophyll A absorbs at blue, and this is drawn by the blue pen, absorbs the most at the blue and the red region. And also this action spectrum graph with other experiment tells us that the rate of photosynthesis is high at blue and red. So from these two, what do you conclude? Any conclusion you can come? The conclusion that you can come is, I am writing the conclusion here in the graph, that chlorophyll A is the main photosynthetic pigment. Yes or no? Isn't this the conclusion that we come? That chlorophyll A is the main. If you superimpose this graph on this graph, this line or this curve superimposes with this curve. When this curve superimposes with this curve, we come to the conclusion that chlorophyll A is the main photosynthetic pigment, isn't it? Because the blue and red light are the most effective lights which affects the photosynthesis the most, and chlorophyll A is the pigment which absorbs the blue and red light. So chlorophyll A is the most effective photosynthetic pigment. Now you understand that why I told that chlorophyll A is the most effective photosynthetic pigment and forms here I have told you, see, forms the reaction center, and other also are accessory pigment, because other all pigments are absorbing light at different wavelengths. It's just, those wavelengths of light are not so effective in doing photosynthesis. It just absorbs the energy and passes it to chlorophyll A. So that I hope you have understood. Okay. So now we will go. So tell me in comment section you have understood this or not. Please tell me in comment section that you have understood, and did you feel this is interesting or not, or you have understood or not. Please tell me. Then I will shift to next topic, that is Emerson's effect. Okay. Okay, now we will shift into the next topic. Okay. Now I will shift into the next topic, and the topic is known as Emerson's effect. Yeah. So Emerson's effect is the topic that we will be doing now. Now Emerson is a scientist who studied the quantum yield. Who studied what? The quantum yield. The quantum yield means the amount of O2 released per quantum of light absorbed. Quantum yield means amount of oxygen released per quantum of light absorbed. Now those who have question about what is per quantum of light, you know from my chemistry classes what is per quantum light, or those are not have other chemistry teachers, please ask your chemistry teacher what is per quantum of light. But I hope that you understand what is quantum of light. Is it or not? So the amount of photo released per quantum of light absorbed is known as the quantum yield. So what he did, he actually measured the quantum yield. Okay, so before going into quantum yield, you see what happens is, let me draw a graph again. Okay, now let me plot feed 400 nanometer to 700 nanometer here. Okay, so let me plot from 400 nanometer to 700 nanometer, and let me plot rate of photosynthesis here. Now the thing you will say, sir, এই গ্রাফটা তো we have seen. Obviously you have seen. But when Emerson did it, Emerson observed that, okay, now what is this happening? This is the main concern. See, absorption of light is there, highest at blue, but just right after red में थोड़ा सा increase होगा, but right after red increase के बाद there is a drop in the rate of photosynthesis. This is known as red drop, and that's why the experiment he conducted is known as Emerson's red drop experiment. Okay, so the experiment he conducted is known as Emerson's red drop experiment. You see, this is Emerson's red drop experiment, and in Emerson's red drop experiment, the graph that Emerson obtained said us that fine, after the absorption, if you measure the rate of photosynthesis, okay, it is going up little bit, the rate of photosynthesis, and okay, it is going up at the red region also, but right after the red region there is a drop. Okay, there is sudden drop in the rate of photosynthesis after 680 nanometer, he observed. There is a sudden drop at the rate of photosynthesis after 680 nanometer. Wow. Fine, if there is sudden drop at the photosynthesis at 680 nanometer, okay. Now these aroused question in him that, okay, fine, it is dropping. Now, what he did is that when he saw that there is drop after 680 nanometer, then he at that range did some experiment. He told that I will measure the rate of photosynthesis or quantum yield, or you can say rate of photosynthesis. I'll measure the quantum yield at 700 nanometer, okay, and I will measure the quantum yield at 653 nanometer. Actually, he measured, at a big range, 600 से लेके 700. We are talking about just two. Obviously, a scientist will not work with just such a big experiment. We will not just work with two wavelengths. He actually worked with all the wavelengths because this drop has, raised question in his mind. Why this drop is there? So, he already knew that এখানে তো বেশি হচ্ছে, fine. But এখানে হয়ে why there is a drop. Now this drop can be explained that, okay, fine, these lights are not so effective for photosynthesis, so here the rate of photosynthesis is low. But why there is a drop right after red? Why there is a drop right after red, or just red reach korei there is a drop? Red may reach kore. Don't think that, sir, red er pore to infrared sejonno drop. No. Red er bhitorei there are red er একটা range আছে. There is a range of the red wavelength or the wavelength which red color এর wavelength একটা range আছে. ঠিক আছে, তো কেন এরকম হচ্ছে যে 680 ন্যানোমিটারের পর ড্রপ হয়ে যাচ্ছে? তো কি করলো? What he did is quantum yield at 700 nanometer he tried to measure, quantum yield at 653 nanometer he tried to measure. He found that quantum yield at 700 nanometer is 10 and quantum yield at 653 nanometer is 53. What is this? Then he understood that, okay, the drop is there because there is saturation. When a chemical reaction occurs, yield spelling dekhe na Snehangshu, why are you after my life? Okay, yield hi hobe. Yield মানে কি? Production. Okay, so anyway, তো ও এটা দেখলো, দেখে বুঝলো যে ও আচ্ছা, তার মানে এখানে স্যাচুরেশন হচ্ছে, এখানে এত হাই রেট অফ ফটোসিনথেসিস 653 ন্যানোমিটারে, ঠিক আছে। কেউ কেউ এটা 680 ন্যানোমিটারও ভাবে, কোনো বইয়ে এটাকে 680 ন্যানোমিটারও দেওয়া হয়। যে স্যাচুরেশন হয়ে গিয়ে এত বেশি প্রোডাকশন হয়, স্যাচুরেশনটা কত কম, 7, তারপরে রেট অফ ফটোসিনথেসিস কমতে থাকে, ঠিক আছে। স্যাচুরেশন হয়ে যাওয়ার জন্য কেমিক্যাল রিয়াকশন স্যাচুরেটেড হয়ে গেলে তাহলে রেট অফ ফরওয়ার্ড রিয়াকশন কমে যায়, এটা কেমিক্যাল ইকুইলিব্রিয়ামে তোরা পড়বি। Okay, you will read this in chemical equilibrium that forward reaction when the products are produced, okay, if the products get saturated or increase in the amount of concentration, then the what we can say the rate of that reaction slows down. Suppose A is reacting with B to give C. C এর concentration যত বাড়তে থাকবে তত A আর B এর কমতে থাকবে, আর if this is a reversible reaction, then what happens is that C ka concentration increase hote hote that this reaction becomes slowly it becomes, tries to attain less. তাহলে এখানে স্যাচুরেশন হয়ে যাচ্ছে, তাই জন্য quantum yield is getting less. Fine. But the most interesting thing that he noticed is that, okay, the interesting thing that I am noticing is what? The interesting thing that I am noticing is the fact that if I do it together, suppose he exposed the plant at 700 nanometer, 10, he exposed the plant at 653 nanometer, 53. But when he exposed the plant at together 700 nanometer and 653 nanometer, now what is this? আমি তো এখনই একটা স্যাচুরেশনের থিওরি দিলাম, তার মানে স্যাচুরেশনের থিওরিটা কি হয়ে গেল? ভুল হয়ে গেল। কারণ কেন ভুল হলো? দেখ, দিস স্যাচুরেশন থিওরি বিকাম রং। সো আই স্যার জানপুচকে আই গেভ ইউ স্যাচুরেশন থিওরি সো দ্যাট ইউ ট্রাই টু আন্ডারস্ট্যান্ড এন্ড নেভার ফরগেট দিস। এখানে আমি কি বললাম? হয়তো হোয়াট ইজ হ্যাপেনিং 653 ন্যানোমিটার আফটার 680 ন্যানোমিটার, মেবি দ্য কোয়ান্টাম yield is so high, the quantum yield is so high, maybe due to this reason of getting saturated of the products, what is happening is the quantum yield is high and saturation, the rate is becoming slower. But if it is so, if this would be your if this would be your conclusion, then Emerson, just made me look fool in front of you. How? Because what he did, he exposed the plant to only 700 nanometer, quantum yield was 10. এবার বল, ওই প্ল্যান্টটা তো আগে ফটোসিনথেসিস করে নাই। He exposed it to only 653 nanometer, the quantum yield was 53. And then what he did, he exposed at 700 and 653 together, means প্ল্যান্টটা ছিল, this was the plant, first 700 nanometer এক্সপোজ করলে কোয়ান্টাম yield 10, তারপর 53 nanometer এক্সপোজ করলে 653 তে বা 680 তে করলে কোয়ান্টাম yield 53। দুজনকে একসাথে করলে, if both the lights are allowed to fall, both the wavelengths together, the quantum yield becomes 72. Now this is something very interesting. Are 10 or 53 to 63 na, then why it is coming 72? Then what actually is happening, that it is most probably কি হচ্ছে যে এরা দুজন একসাথে কাজ করলে, means in the thylakoid or in the chlorophyll pigments, some chlorophyll pigments are absorbing 700, some are absorbing 653, and both of them when together they are working, giving rise to 72 quantum yield, means together when they are working, they are giving the largest amount of yield. তার মানে if you are exposing it to 700 nanometer, the other one which is supposed to work at 653 nanometer or 680 nanometer is not working, has stopped working. That's why your quantum yield is less, that chlorophyll pigments. And if you would have given just 653 nanometer, then quantum yield is okay, because the molecule, this molecule which is absorbing, or this chlorophyll molecule which is absorbing the maybe A, B, C, D, whatever, this chlorophyll molecule or the particular molecule or the particular structure which is absorbing the 653 nanometer is working alone, hence giving 53 and not getting the support of 700 nanometer ka molecule, the molecule which is absorbing. তার মানে মোদ্দা কথা হচ্ছে যে এটা যদি এখানে হয়, অনেকগুলি क्लोरोफिल पिगमेंट এরকম আছে, অনেকগুলি बहुत सारा क्लोरोफिल पिगमेंट ऐसा है जो कि 653 या 680 एब्जॉर्ब कर रहा है, बहुत सारा क्लोरोफिल पिगमेंट ऐसा है जो कि 700 एब्जॉर्ब कर रहा है, और दोनों मिलके काम करेगा तब क्वांटम यील्ड हाई हो जाएगा। अगर आप सिर्फ 700 दे रहे हो तो ये बेचारा काम ही नहीं कर पा रहा है, और अगर 653 दे रहे हो तो ये बेचारा काम ही नहीं कर पा रहा है। इसी वजह से रेट ड्रॉप हुआ है इस जगह पे आके, ठीक है, वहां पे रेट ड्रॉप हुआ है। क्यों? क्योंकि रेट के बाद अब्सॉर्ब करने लायक कोई फोटोसिंथेटिक पिगमेंट ही नहीं है। जो पिगमेंट्स थोड़ा सा रेट के बाद, और जो मॉलिक्यूल्स, वहां पे तो बहुत सारा मॉलिक्यूल फैला हुआ है, थाइलाकॉइड के अंदर मैंने जैसे सिखाया बहुत सारा मॉलिक्यूल फैला हुआ है, राइट और रॉन्ग, क्लोरोफिल मॉलिक्यूल्स। सो उससे ज्यादा वाला जो अब्सॉर्ब करेगा वो लोग फोटोसिंथेसिस को उतना हाईली करा ही नहीं पाता है। आर यू गेटिंग माई पॉइंट? सो दिस इज व्हाट इट इज। तो यहां से वो और एक कंक्लूजन में आया। नो, टेल मी इन कमेंट सेक्शन, डिड यू अंडरस्टैंड? Tell me in comment section, did you understand? It was clear, Obirup, right? Okay, good. Now, there is photosystem 1, there is photosystem 2, which we'll learn in the next video. But this thing you keep in your mind. Till then, thank you. And one more thing, let's do the 10:30 ka thing tomorrow, okay? Not today.